Hi, everybody. So thanks for coming. So my name is Stuart Lichtenberg. I'm a scientist here at the University of Minnesota. I'm an environmental toxicologist by training, but lately I've been studying something known as chronic wasting disease. And you might be wondering, well, what exactly is an environmental toxicologist doing studying an animal disease? That'll all become clear as we get into the presentation. And let me just go ahead and share my screen. We'll get started. There we go. We're showing up loud and clear. Good.
Yep.
Excellent. So today we're going to be talking about chronic wasting disease, or CWD. I'm going to give you all a little bit of background on that. We're going to talk about prions, what prions are, and why both of these matter to the real estate industry. That's obviously the reason we're all here, and the answers may surprise you a little bit. So first, I wanted to go over, a general overview of what we're going to be covering today. First, we're going to talk about what chronic wasting disease actually is, why it happens, what it's made of, so on and so forth. We're going to talk about what prions are. You'll hear this word come up repeatedly throughout the course of this presentation, and it's kind of really important to drive home what exactly prions are and why they're different than most other pathogens. We're going to talk about these prions' behavior in the environment. This is where the real estate angle sort of comes into things. What do prions do in the environment? Where do they go? So on and so forth. And lastly, why does any of this matter to realtors? Why is a environmental toxicologist and biologist talking about animal diseases? Well, that'll all become clear as we move along. So first things first, let's tackle the first question here. What exactly is chronic wasting disease? Chronic wasting disease is what we call a transmissible spongiform encephalopathy. It's a long word for a brain disease of deer. It causes a number of different symptoms throughout the course of the disease, the most obvious of which takes place in the name, wasting. So you'll see this image of this deer right here, probably fairly late into the disease, and it's showing kind of the hallmark sign of the disease. It's emaciated. You can see its spine and its ribs quite readily. Another sign of chronic wasting disease is increased thirst and drooling, both of which you can see in this photo as well. Here's a picture of an elk that's also come down with the disease. This is kind of a classic photo from one of the scientists that first discovered CWD. You can see it's drooling quite heavily. So what chronic wasting disease ultimately leads to is a degeneration in the brain of an affected animal. Brain stops working quite as well. It develops all these symptoms. Ultimately, the animal will die of the disease. It experiences some selective neurodegeneration, not unlike Alzheimer's or Parkinson's disease in humans. And one of the key features of chronic wasting disease can also be found in that name that I mentioned earlier, transmissible spongiform encephalopathy. It's transmissible. It can move from animal to animal. How it moves from animal to animal isn't quite clear yet. We have a lot of ideas. We have a lot of theories about ways that it can, but in terms of any definitive way, we haven't quite nailed that down yet. As it is transmissible, it's also spreading. So this is kind of what we call the United States CWD map. This is produced by some folks over in Madison, Wisconsin. This map is on probably at least half of the presentations that anybody ever gives on CWD because it does such a great job of illustrating why this is such a problem across the country. So this is the most recent map taken in 2020, created in 2023. Each of the little gray counties are areas where CWD infected deer have been detected in the wild. Each of the yellow dots are captive facilities that have been depopulated. So a captive deer farm where CWD was detected and then the animals were removed. Red dots are captive facilities that currently have animals on them still. So it still has live infected animals being present. And you'll notice over in the area of Colorado and Wyoming, there's some darker gray. That's where CWD was originally detected in the wild prior to the year 2000. So all of this growth that you've seen, that you can see on the map, aside from those dark gray areas, has happened in the last 23 years. So it's spreading fast, and it's spreading all over the place. You'll notice in particular, we do have a few spots in Wisconsin where it started to pop up. This is a relatively recent phenomenon. It's been spreading for quite a bit longer in some of our neighboring states, particularly Wisconsin. But it is here, and it is slowly increasing in prevalence. For instance, here's a map of Minnesota that shows the different CWD management zones as dictated by the Department of Natural Resources. So each of these areas have different regulations related to deer hunting and carcass management and certain other restrictions as well. And these are the areas where we're mostly finding it in the wild. So these are the areas that are principally affected by chronic wasting disease. But again, because of the way the disease behaves, you can pretty much count on this map getting bigger over time. Inevitably, we will get more detections, and it will spread more and more outside of the areas that are shown here. But this is where we're at right at this time. So in order to really get a picture as to what CWD is and how it behaves and why it behaves the way it does, we need to talk about things called prions. So what is a prion? So a prion is a pathogen. It's a thing that causes disease. Now, pathogens you may have heard of before pretty commonly are things like viruses, shown to the left, bacteria, shown at the right. Fungi cause disease too, but I didn't have time to put them on this slide, but we'll count them as well. Prions are pathogens, but they're entirely different. They are neither viruses nor bacteria. They are completely distinctive compared to any other known pathogen out there. Now, why is that, and what are they made of? To understand that, we need to go back to our old Biology 101 class and talk about how information moves in a biological system. So most folks have probably heard of DNA. I'm hoping you can see my cursor. DNA are the blueprints for all of the different biomolecules that we and all other living organisms have. The general way things move is DNA makes a molecule called RNA, which then serves as kind of a temporary template to make proteins. You'll notice the information flows from DNA to RNA to protein, never the other direction. There's never information coming from proteins that influence DNA or RNA, likely lead to the creation of information by proteins themselves. Now, there are some other things going on here. We're going to gloss over those for the time being. The important thing is to know that protein typically is sort of the dead end in terms of biological processes. It's proteins that actually do things. So let's talk about what proteins are and what they do. So when we're talking about a protein, in the simplest sense, it's a linear polymer with a series of different molecules. We call those amino acids. This primary structure shown up here at the left is kind of an example of how a protein might look. Each of these little circles represents a different type of molecule that goes into building a protein. And different proteins have different arrangements of that. But ultimately, they're all long linear chains of amino acids, one after the other, just a long string of them. So you can think of it in its primary structure as just a long line of amino acids. Now, depending on how those amino acids in that structure interact with themselves, we can actually get what we call secondary structures that develop. And these are shown down here at the bottom. We'll call these helixes and sheets. So helixes, you can usually see these represented on diagrams as these kind of tight little spirals like that. And sheets are usually have these more ribbon structures right here. So each of these can come about as a result of secondary structures. We can also get into things we call tertiary structures, which is how the entire protein molecule arranges itself in 3D space. So it can be a combination of helixes, sheets, or neither, but it's just how the protein looks once it's all assembled and taken its shape. We can also have things called quaternary structure, which is when more than one individual protein associates with another individual protein. This might be a different type of protein, or it might be the same type of protein, but it's basically more of them associating together. And all of this basically results in little biological machines. Proteins are things that do things one way or another, and that's all entirely dependent upon this three-dimensional shape. A good analogy that we like to use here at MinPro is slinky. You can think of a protein as a slinky. It's a linear, just one long chain of something. And more importantly, to what we're talking about here, slinkies do something. They go downstairs, for instance. When we're talking about prions, though, prions are pathogens that are made solely of protein. Now, you can think of a prion as a slinky that has gotten kinked up. It's gotten twisted on itself, so it no longer does what it's supposed to do. Prions are exactly the same concept. They are a normal protein that has taken on a shape that is distinctly different than its original shape that does the job that it's supposed to be doing. And there's actually some distinct features of prions that we know about. So this diagram right here demonstrates one of the first things that was picked up on prions as pathogens. So we see here, this is a normal prion, this PRP with the C right up here. You'll notice there's a whole lot of these helixes present. Well, we were able to determine over a series of years that the prion protein, when it takes on this pathogenic shape, this incorrect shape, this misfolded shape, or twisted slinky shape, we get a whole lot more of these sheet structures. What happens over time is these sheet structures, these misfolded prions, accumulate in cells and ultimately lead to cell death. Now, another key feature of these prions is that they're able to template the conversion of healthy prion into misfolded prion. So this diagram at the bottom kind of conceptually lays this out. We have our diseased prion right here, which can actually interact with healthy prion protein and turn it into this shape right here that's a diseased shape. Now, ultimately, what happens when this occurs is that these prions tend to stick together. They form what's known as aggregates. They end up absolutely huge in the biological sense. And because they are so big, other proteins that are designed to break down proteins no longer act upon them. They no longer get broken down in normal biological systems. They tend to just accumulate in cells, particularly in neural cells associated with like brains and neural tissue, ultimately interfere with all kinds of other cellular processes and lead to cell death. There's actually some work that's been done recently that's given us a really good idea of what prions actually look like. So this is an electron microscope image of some actual prions. Little rod-like structures right here that you can kind of see in the gray right here. Those are actually prions. Those are what we call prion fibrils. These are only 100 nanometers long, which is very tiny, but in the world of cells and proteins is actually enormous. There's even been some work that's actually looked at what prions really look like in terms of how the amino acids stack on each other. And that's what we can see right here in this image. You can see each one of these colored ribbons right here represents a single prion molecule stacked one on top of the other, building that fibril that ultimately builds up in animals' bodies and leads to all of these neurological problems. So there's one other distinctive feature of prions that we need to talk about before we start really getting into how this influences prion behavior in the environment and why it matters to real estate. And that is, over the course of the disease, infected animals shed prions. This can happen in a few different ways. So one way is in urine. Right here, we have a deer peeing on the landscape. This deer had chronic wasting disease. There's a distinct possibility that he could be releasing prions into the environment via his urine. It can also be spread through feces. So over the course of the disease, prions are shed in animal feces. These are deposited onto the landscape as well. Another way is through saliva. So shown here is something that we call a mineral lick. You can see this kind of indentation in the soil right here in the center of this image. This is where deer have actually gone and licked up all of that soil and left these large stones behind. We actually have photographs of where they've managed to tip over trees by uprooting them from just digging into the soil. And when they're doing this and eating all these minerals, they deposit saliva, which also contains prions. And kind of the last big way is just through animals dying. When an animal dies and its various organs or other materials are left on the ground, all of the prions that are retained in those tissues are released into the soil as well and into the broader environment. This also comes about through things like hunting, gut piles, for instance. The CWD positive animal, the gut pile is left as a result of that. Any prions left in that will be released into the environment as well. So more to this, prions shed into the environment end up remaining infectious. So even though those prions have been released and they're sitting around in the environment, they can still sicken other animals. So if, for instance, an animal was to come across a spot where one animal had licked or peed and interacts with that in the right kind of way, either through ingesting the soil, most likely, or some other way, it's quite plausible that that animal could get sick from interacting with that landscape that had been previously contaminated. Perhaps most alarmingly of all of this is prions remain on the environment for a long, long time. So I mentioned earlier about how normal biological processes don't break down these infectious prions. Well, if you can think about it this way, if normal biological processes that are occurring in us don't break down prions, biological processes occurring in the broader environment aren't going to break them down either. So things like bacteria or funguses in the soil aren't going to break down these prions at appreciable rates. The record so far in terms of prion disease persisting in the environment is 16 years. I've been working on a project actually that has evidence that we can have prions persisting from chronic wasting disease for 15 years. So this is just a little bit of work that we've been doing here at the university. This is some work that we did at a deer farm that had been contaminated in the early 2000s. Ultimately, it ended up being depopulated. This is a view of the landscape here. You can kind of see the area where the deer would interact with the soil over here, run along this fence line. You can see also right here where lots of animal traffic occurring. At the time of depopulation, there were a number of animal carcasses on the site that had actually passed away from the disease as well. So fortunately for us at the time, there were some soil samples taken from this site around the time that farm was depopulated. These were stored, and then there was an attempt at decontamination on this site. So the state agency responsible for the site scraped off the top 6 inches of soil off of this farm and then sprayed everything down with bleach. It's a fairly thorough effort to decontaminate the site. You can see here another fella spraying various places, trying to just get all of the site. Well, we were able to actually go back in 2021 and collect soil from that site as well and compare it to the original soil. And the short answer is that prions are still present at this particular site, 15 years after this attempted decontamination. It's a little bit less than what we had seen before, but it's still there. Fairly certain that if we were to reintroduce animals onto this site, they would ultimately get sick. So this gets into why this is important for real estate. Obviously, if we have infectious agents that can be deposited on the landscape, it becomes a concern for folks who are tasked with dealing with land issues, such as realtors, for instance. So there's been some changes to Minnesota law that have taken place over the past year that are quite salient to real estate as related to captive service operations. So this was a law that was passed this year, and it's a modification to these Minnesota statutes right here. If anybody has any questions about this at any point, just let me know. I can send the actual full text. I've abbreviated this to some extent to focus on the points that are most relevant for you all. So in this case, this subsection is amended to read variety of things about possessing live cervidae, which is another word for deer or elk, so on and so forth. The first point is that the board is not allowing new registrations of captive cervid farms in the state of Minnesota. Essentially, nobody is going to be allowed to open a new captive cervid facility in the state. So anybody purchasing land with the intention of opening such a operation should be dissuaded as such. They're not going to be permitted to do that sort of thing any longer. There are provisions, however, that an existing farm can be sold or transferred once under this law, but it must be to a family member, and this can only happen once. After that, it's not allowed to be transferred again, and it has to be on existing property. There's another section that has some relevant points to real estate too. The next section, subdivision 11. This relates to farms where chronic wasting disease has been detected. So if a landowner at any point who is operating a cervid farm on their property is detected, there's a series of things that must happen with them contacting the Board of Animal Health and Natural Resources Office. But the big ones that are important related to real estate are this. Before signing an agreement to sell or transfer the property, the owner basically has to disclose the date of depopulation, in other words, when the animals were removed, and the requirements upon the premises. Typically, in these cases, fencing has to be put up on the site to secure it, to prevent other animals from entering the site once CWD has been detected on the site. This can be quite expensive, so worth informing potential buyers about this sort of thing. Lastly, landowners must also inform the county registrar that they have been operating a deer farm that has been detected as CWD positive. So this is, again, contingent upon the landowner to do this sort of thing, but something worth knowing as a realtor. Now, this is the salient points that have been in the law as it is right now. Now, there's some important points to discuss of what's not in the new legislation. An important point here is that so far, this legislation largely concerns captive deer farms. So this does not apply to things such as hunting properties. So something that would be undeveloped land with free-ranging deer present. There's no obligation for a landowner to inform the county registrar that their property has specifically had CWD detected. There are provisions that the DNR will probably take care of, but it's nothing that's going to be announced publicly, and it's not anything that requires the landowner to take any steps outside of what they would normally be doing. As such, there's no provisions for reporting CWD status of animals taken from a non-farmed property. So that's the important distinction right here. As of now, there are no requirements specifically on landowners if they're not operating a commercial deer farm. That's the big distinction right there. So I wanted to leave plenty of time for questions. I'm hoping we've got some that have come up, but I wanted to take a moment to review real quickly. So we talked about chronic wasting disease, how it's a prion disease that affects deer, elk, and moose. Prions are shed into the environment by sick animals over the course of the disease. And probably the most important here is prions are not like other pathogens. They stay in the environment for years and they remain infectious. And lastly, we've had some new laws that have been enacted, which specify certain actions that must be taken when cervid farms are bought or sold. And I just wanted to take this time to make you all aware of it. And with that, I'll open it up to any questions that anyone may have.